Okay, class, we're going to finish out this chapter on genetics by covering a very topical subject, viruses. Viruses are submicroscopic infectious agents. They're smaller, much smaller than cells. And essentially, they are a nucleic acid which could be either DNA or RNA, which could either be single-stranded or double-stranded. They're a nucleic acid encapsulated by a protein coat and usually also encapsulated by a lipid membrane. So they're a piece of nucleic acid which codes for the proteins needed by the virus to survive. Now, I say survive because there is a question as to whether or not a virus should be considered to be alive. I think most biologists would not describe a virus as being alive, but instead that they are on the edge of life. A virus cannot reproduce by itself, one of the chief requirements to be said to be alive. They cannot reproduce by themselves. They must have some host, and we'll talk about their life cycle or how they reproduce, but they must live within a host at some stage, and they have no metabolism themselves. That is, they do not extract energy from any fuel themselves. The virus is actually similar to other small pieces of nucleic acid that sort of move between cells. These are called plasmids and transposons, which we won't have time to talk about. But viruses seem to have been around almost as long as the first living cell. Now, the way viruses work in general is that they invade a cell, and that cell could be either a bacterium cell, a plant cell, or an animal cell. And certain viruses actually will use specific types of host. That is, they might use only a plant or only a bacteria or only a human or only a pig as a host. They invade the cell of the host and they take over the machinery within the cell. In the case of eukaryotic cells, they will take over the ribosomes and the tRNA and the sources of energy, the ATP and the other nucleotide triphosphates, in order to make more copies of themselves. They essentially invade a home, take over everything inside the home, raid all the refrigerator and all the fuel sources, make more copies of themselves, make more babies until the cell essentially will lyse or will rupture in many cases and spill out more copies of the virus, which then go around to infect other nearby host cells. Now, our immune system will usually respond by making antibodies, and we'll talk about that in a second. But in some cases, the virus becomes latent in a sense that it will embed itself into the chromosome, the DNA chromosome of the host, and essentially hide, and maybe hide for a long time. And that's one of the reasons why it's difficult to treat some viruses. Some of these viruses that become latent include the HIV virus and hepatitis virus. The problem with an infection by a virus with we as the host is that not only does it rupture and kill cells, but it oftentimes leads to a lot of other symptoms that lead to diseases, and in some cases can be fatal diseases. Now, viruses have been with us for a long, long time. But of course, mankind didn't know what to make of them or what they were. One of the first scientists who had a clue about a virus was Louis Pasteur, who was studying rabies, and he found that there was some infectious particle that was so small that it could not be trapped on a filter. And so he says, there must be some kind of very, very small infectious particle. In addition to Pasteur studying rabies, one of the next scientists who began to study viruses was a Russian scientist, Dmitry Ivanovsky, in about 1892, and he was studying the infection of tobacco plants. And he discovered a particle, an infectious particle called the tobacco mosaic virus, TMV. Within the next decade or so, he and other scientists began calling these a virus, and they began determining a little bit more information about them. The tobacco mosaic virus, which is shown on this slide, is an interesting structure. It is made of a single-stranded RNA nucleic acid, which is coated by a single protein. So it's a rather simple virus, and it looks like a spiral staircase, as you can see. Now, that's an example of a virus that will infect a plant. A number of viruses infect bacteria. And in fact, it's estimated that the most predominant individual type of bioparticle in the world are these bacteriophages. There are more copies of them than anything else on planet Earth. But of course, they're very, very small particles. This is the structure of a bacteriophage. And as you can see, they can be very elaborate, interesting looking structures. Almost looks like something that's supposed to land on the moon or on Mars, where near the top you will have a capsid, sounds like a moonlander, with the nucleic acid, in this case DNA, and you will have other parts that will bind to the plant and actually this acts almost like a syringe or an injector. It will inject the DNA into the cell, and where it then takes over that machinery and makes copies of itself. So that's an interesting structure for a bacteriophage. But now on to viruses that infect us. The most common one is the influenza virus. So when you say, I've got the flu, you've probably got influenza type A or some variant of influenza A flu. The influenza virus is a RNA virus, meaning that in the middle of it, it has a RNA molecule, ribonucleic acid, and it's surrounded by a lipid coat and proteins that are sort of attached at the surface of this membrane coat. So this is the influenza virus. We should receive an annual vaccination for the influenza virus, and as you may know, sometimes you have to get a different vaccination each year because the virus mutates. We'll get to that a little later. One of the more serious outbreaks was the 1918 Spanish flu, which was a variant called the H1N1. Now, there are a number of variants of flu that we oftentimes identify by these letters H1N1, or it could be H2N1, or H5N1, or H1N2. And this has to do with some of the proteins like neuraminidase and hemagglutinin. So the N and the H, different variants, different versions of these proteins on the surface, and they then will elicit different immune responses. But the Spanish flu of 1918 was one of the most deadly outbreaks in the history of mankind. Caused about 50 million deaths worldwide and about 675,000 deaths in the United States. More recently, in 2009, the influenza virus, again the H1N1 variant, caused about 500,000 deaths worldwide and about 12,500 deaths in the United States. So influenza itself can be deadly. It's serious, and we have it every year, and we should be getting vaccinations against the influenza virus. Another virus that you may have heard of is the herpes simplex virus, HSV1. There's also an HSV2. HSV1 is commonly associated with cold sores, and HSV2 with genital herpes. This virus is a DNA virus, has DNA as its nucleic acid, and again, it's coated with a lipid coat with these proteins, these envelope proteins that are on the outside, which sort of bind to various human cells. Now, the interesting thing about herpes simplex virus is that it is one that shows this phenomena of latency. That is, it can become incorporated into the chromosome of our cells, and particularly our nerve cells, and stay hidden for long periods of time before it is expressed. And that's why people who are infected with herpes simplex virus will sometimes have cold sores. They'll go away, they'll come back, go away and come back. And just to let you know how prevalent it is, it's estimated that about 2/3 of people below the age of 50 are host to HSV1 virus. So about half of you will have this virus in a latent stage in your bodies. Now, it's not expressed by all of us, or it doesn't appear as with symptoms, but a lot of us have it. Now, another virus that is very, very serious is the HIV virus, which of course we associate with the disease of AIDS. The HIV stands for human immunodeficiency virus, and AIDS stands for acquired immunodeficiency syndrome. Now, AIDS is an RNA virus. You'll see that in the center it has RNA, and it has a double coating. It has an internal capsid made out of proteins, and then it has a lipid membrane surrounding it, and also these coat proteins that are on the outside. So many of these viruses that infect humans have somewhat of a similar structure, the variance being whether or not they're RNA or DNA inside. Now, the cycle for an HIV virus is that it will first bind to a cell. Now, some of these proteins on the surface, one's called GP120 and GP41, these are called glycoproteins, will bind to receptors on a cell. This would be the larger cell that's being infected, will become engulfed by a process we call endocytosis, bringing into the cell. And then the membrane of the capsid dissolves away, and the nucleic acid, the RNA, is converted to DNA. So viral RNA is converted to viral DNA using an enzyme that the virus brings along with it, that it already has, called reverse transcriptase, which we will abbreviate as RT, reverse transcriptase. It catalyzes the copying of the genetic information of the virus from its RNA format to a DNA format. Remember the central dogma of molecular biology. We usually go from DNA to RNA to proteins, but here we're going from RNA to DNA to proteins. And we said that that first step could go back and forth. Now, after we make a DNA copy of the viral information, or after in our infected cells, it then enters the nucleus of a cell. This is the nucleus, and that viral DNA becomes integrated with a chromosome of the host cell. And there is an enzyme called integrase, which is important for catalyzing this integration into the host chromosome. Now, at some time later, this chromosome is expressed, and we begin synthesizing proteins for it, including the viral proteins. And that's what happens. And the virus then makes a copy of itself. Now, there is also some processing of the proteins, that the proteins of the capsule of the virus. There's some processing of these by enzymes called proteases, or proteolytic digestion, or proteases. Now, I say these steps because you may know that with treatment of AIDS, oftentimes a person who has AIDS is given a cocktail of 2, 3, or 4 different drugs, and these drugs are trying to block or inhibit the action of the AIDS virus at different steps. That is, either at the binding step, or inhibitors of reverse transcriptase, or inhibitors of integrase, or inhibitors of these processing proteases. So this is how much we understand about the AIDS virus. Now, this AIDS virus, it turns out, we'll talk about a little bit later about how many deaths, is probably the most serious cause of death over the last 40 years of any of these types of infectious diseases. There have been 30 million deaths over the past 40 years, including about 675,000 deaths in the United States of victims of AIDS. Now, the one that's in the news these days, of course, is COVID-19. Coronavirus 19-4, its discovery in 2019. It is sometimes called SARS-CoV-2, and that's because it is very similar to a related coronavirus, an outbreak of which occurred in 2002 to 2004, called SARS-CoV-1, severe acute respiratory syndrome. That's what SARS stands for, severe acute respiratory syndrome. And another coronavirus called MERS-CoV, which MERS stands for Middle East Respiratory Syndrome. And that's because this latter one was discovered in the Middle East and actually is confined to the Middle East. And that outbreak was in 2012. Now, these first two coronaviruses, while they were detected and they were somewhat serious outbreaks, there were no deaths reported in the United States attributed to these, and there was less than 1,000 worldwide deaths associated with with SARS-CoV-1 or with MERS. But of course, with SARS-CoV-2 or COVID-19, the story is a whole lot different because it's a very infectious coronavirus and fairly lethal as well. At the time that I'm making this slide, we're approaching about 140,000 deaths in the United States and about 3 million cases detected in the United States. Now you might wonder, well, since these coronaviruses were first detected, in 2002, 2003, do we have vaccines against them? But unfortunately, they were not able to successfully make vaccines against either the first SARS-CoV-1 or MERS. So let's knock on wood that they'll be successful with COVID-19. Now let me talk about some other very deadly viruses which you may have heard of. One is Ebola virus. You've probably heard of Ebola, for which there have been outbreaks in Western Africa. It's a very fatal virus. Its fatality rate is about 50%, even when treated. There have been about 11,000 deaths in Western Africa associated with this virus. Fortunately, there have only been one death so far that has been documented in the United States, and only 4 patients. 2 of these were doctors, physicians who came back from West Africa after treating some of the victims in West Africa, and then 2 nurses that treated these physicians here in the United States, and only 1 out of those 4 died. But it is a very deadly virus, and it leads to sort of breakdown of the tissues throughout the body and bleeding through all pores. So it's a pretty deadly virus. A couple of others that you may have heard of the Marburg virus, the hantavirus. These are also very deadly viruses. The Marburg virus, about 90% fatality. It's spread by bats. The hantavirus spread by rats. It's about 40% fatal. And that gets us back to rabies, which we talked about earlier, which has been known by mankind essentially forever. It's spread by being bitten by dogs or bats or squirrels, and it leads to an inflammation of the brain. It can be almost 100% fatality if not treated, but fortunately we have treatments for rabies. In this last slide, I point out that viruses are generally not treatable by antibiotics. Antibiotics are small molecules that can either be made by scientists in laboratories, by bacteria and fungi, as ways to fight off bacteria. But antibiotics in general do not work against viruses. Now you'd say, well, how about antiviral agents? It sounds like that might be something similar, except it's working against the viruses. Well, there are a few that may have heard of, like Tamiflu or Remdesivir, which are antiviral agents. But here's the problem with fighting against the virus. The virus invades the host cells, that is like our cells, and uses our own machinery, our own ribosomes, our own proteins, our own enzymes. And so an antiviral agent has to work against our own cells. Find an antiviral agent that will work specifically against an enzyme used only by the virus, which is part of the trick. Also, viruses can mutate very frequently, and that leads to the problem that if we do have a vaccine, and a vaccine is about the only good way to treat a virus, if we do have a vaccine, we oftentimes have to be vaccinated again every year to be immune, or we can even be reinfected by a virus because that virus may have mutated, and our own immune system may only remember how to mount a immune defense against the original version of the virus that we were infected with. Now, while saying this, vaccines can be very effective, and some diseases have been almost completely eradicated by vaccines, and these include polio and mumps and measles, rubella, smallpox, and hepatitis B. Okay, I'll leave you with that, and we've now finished the chapter on genetics and related topics, and there will be a little quiz, and then you will have access to study.